Welcome and good afternoon to today's webinar, PFAS Effects on the Immune System. What do we know and what are the links to COVID-19? My name is Michelle Dixon and I am the Program and Development Director of the Massachusetts Breast Cancer Coalition. And I'd like to thank you all for joining us today. I am very pleased to welcome back today's speaker, Dr. Laurel Schaider. Dr. Schaider is a research scientist at Silent Spring Institute, where she leads the Institute's water quality research on PFAS and other contaminants of emerging concern. She studies PFAS chemicals in drinking water, consumer products, and food, drinking water contamination from septic systems, and environmental justice disparities in drinking water quality. She is the lead investigator of PFAS REACH, a study evaluating PFAS immune toxicity in children and addressing the needs of communities affected by PFAS water contamination. She is part of the University of Rhode Island's STEEP Superfund research program and co-leads community engagement activities on Cape Cod. Dr. Schaider is also the lead investigator of a study funded by the Centers for Disease Control and Prevention and the Agency for Toxic Substances and Disease Registry on PFAS health effects in 2 communities in eastern Massachusetts. Before joining Silent Spring, she was a research associate at Harvard T.H. Chan School of Public Health. Dr. Schaider earned her master's and PhD in environmental engineering at the University of California, Berkeley, and has a bachelor's degree in environmental engineering science from MIT. Dr. Schaider, welcome back.
Thank you very much, Michelle. It's great to be back today. Thank you very much to Massachusetts Breast Cancer Coalition for hosting today's webinar, and thanks to all of you for tuning in. Today, I will be talking about PFAS effects on the immune system. What do we know, and what are the links to COVID? So I will start out with an overview of what are PFAS. My guess is that many of you have tuned in to some of my previous webinars here for MBCC. So I won't go into a lot of background today. I'll provide some. And if you'd like more information, I would recommend you visit MBCC's webinar archive and tune into some of the past webinars where I've talked in greater length about PFAS. Today, I'll talk more specifically about PFAS effects on the immune system. And I will preface this by saying I'm not an immunologist. I am not an immunotoxicologist. My background is in environmental chemistry and exposure science, and I come at this from the perspective of understanding how we're exposed to PFAS and what the potential health effects might be. COVID-19 is on everyone's mind these days, and it's a very new condition. So we don't have definitive answers about whether there are links between COVID-19 severity and PFAS exposures, but I'll talk about some of the reasons why scientists are concerned about these potential links. So when we began studying PFAS at Silent Spring Institute in 2009, these were not making headline news every day, but things have changed a lot over the past decade, and now every day all around the world, PFAS chemicals are making headline news. This is a Boston Globe article from last month, and it talked about kind of an exciting announcement here in Massachusetts. The Massachusetts Department of Environmental Protection finalized our state's drinking water standard. They established a standard of 20 parts per trillion for the total amount of 6 PFAS in drinking water. Massachusetts is one of several states that have taken action to regulate PFAS in drinking water because the EPA has not set a standard for any PFAS chemicals. Right now, there's a federal health advisory level of 70 parts per trillion for the total amount of 2 PFAS chemicals. Many scientists and regulators don't think this guideline is strict enough, and it's not enforceable. So water suppliers don't have to test under federal law. But this Massachusetts standard is among the strictest in the U.S. So PFAS aren't necessarily a household name, but products containing PFAS are familiar to all of us. We can think of Stainmaster and Scotchgard coated carpets and upholstery, Gore-Tex jackets, Teflon pans, certain types of ski waxes and floor waxes, grease-proof food packaging, such as certain fast food wrappers and microwave popcorn bags, dental floss like Oral-B Glide, paints, And I'm always learning new uses of PFAS. The one that I learned about most recently was coatings on certain guitar strings. PFAS stands for per and polyfluoroalkyl substances, which is quite a mouthful. The terminology of PFAS is pretty complicated. I'm showing here two molecules, PFOS and PFOA, which is sometimes called C8. These are two of the most common PFAS that show up in drinking water and in people. That blue zigzag you see in the middle is a carbon backbone. And so PFAS are characterized by having many carbon-fluorine bonds. This makes PFAS extremely persistent, and in fact, they're called forever chemicals because they simply don't break down in the environment or in our bodies. They have some unique chemical characteristics. They have one end of their structures that makes them both water and oil insoluble, so they have water and grease repellent properties, but they also have a water-soluble part that means they can be very mobile in the environment. For those of you who like to think about chemistry like I do, you might notice that the structure looks a little bit like a soap molecule too. And PFAS are used as surfactants, for instance, in a class of firefighting foams that I'll mention in a minute. And they also have some similarities to fatty acids, and it's thought that that chemical similarity is the reason why PFAS can affect cholesterol and other components of our metabolism. PFAS are a complex family of chemicals. They all share some characteristics, but there are literally thousands of different PFAS. When I first made this slide, there were over 4,700 on the list that people were using. But the EPA has an updated list, and now they say there's over 9,000 different PFAS chemicals. And because of their unique chemical properties, they're versatile for many different uses. So how are we exposed to PFAS? Well, PFAS are not naturally occurring chemicals. So they all come from a manufacturing plant like DuPont or 3M. They can enter the environment in several different ways. So from the industrial facilities where they are manufactured or incorporated into products, they can get into the environment directly through air emissions or water emissions or disposal of sludge from those industrial facilities. The PFAS can enter the waste stream and get into the environment from landfills or from discharges from sewage treatment plants or septic systems. And PFAS are also used in a class of firefighting foams called AFFF or Class B foams. These are used for flammable fuel fires. So they're used a lot at military bases, airports, and other fire training areas where you have flammable fuel fires or for training activities. And this is one of the most common ways that PFAS get out into the environment. So once PFAS are in the environment, and we know that they're everywhere in the environment, even in the far reaches of the Arctic, they can accumulate in polar bears and other Arctic wildlife. We can be exposed through a number of different pathways. So the one that I think about a lot, and a lot of other people consider, is drinking water. PFAS can get moved through groundwater and through surface waters and enter our drinking water supplies. There are particular concerns about exposures to infants because PFAS can be passed along during pregnancy. They can cross the placenta. They also can accumulate in breast milk and be passed along to breastfeeding infants. We can also be exposed to PFAS from food. That could be from the food packaging or from the food itself, plants and animals we're eating, accumulating PFAS from the places where they are grown or raised. We can also be exposed to PFAS from consumer products in our home or workplace. And these exposure pathways are not all that well characterized, but there are several pathways that these can enter our body. So one is through the air. Some PFAS are volatile, which means they can actually be in the air. We can breathe them in that way. PFAS also can end up in dust in our homes, and we all ingest or inhale some amount of dust every day, and that's a way that we can be exposed to flame retardants and lead and other chemicals as well. There's also concern about dermal absorption. So PFAS are used in certain cosmetics, for instance, so there's concern that there might be absorption through the skin as well. So I won't talk too much about drinking water today, but there's a lot of concerns about the prevalence of PFAS in drinking water supplies across the U.S. This is a map that the Environmental Working Group has continued to update, and this number of contaminated sites increases every time I look at their map. So that by their latest count, there are over 2,000 contaminated sites in every state, I guess, except Hawaii. One of their most recent estimates was that over 110 million Americans had PFAS in their drinking water. And just last week, I saw that they had updated that number to over 200 million Americans. Now, when you look at this map, you see that there are some states that have a lot of blue dots. And you might think that that means that they are especially contaminated sites. And to be sure, these are states that have some contaminated sites. But what you're seeing here reflected in the density of these blue dots is that some states are taking the lead and moving ahead of the EPA by requiring water supplies to do more testing than is required by the federal EPA. And in states where you don't see very many dots, that might just be a reflection of little testing. Okay, so here in Massachusetts, the Massachusetts Department of Environmental Protection has been requiring some water suppliers to do testing in advance of the new standards that went into effect this month. So far, there are 30 public water supplies that have tested at above 20 parts per trillion. Some of these were discovered a number of years ago and have since been addressed through water treatment or through turning off contaminated wells. With the new standard that's currently in place, it's likely that there will be more discoveries of PFAS in drinking water. And all of this is concerning because the prevalence of PFAS in our environment and their extreme persistence means that virtually all Americans have detectable levels of PFAS in our blood. And this is based on results of nationwide testing by the Centers for Disease Control and Prevention. At the beginning, I showed you a picture of PFAS and PFOA. They're examples of what we call long-chain PFAS. So they have that long blue carbon backbone. And because of their extreme persistence in people's bodies, these are no longer manufactured in the U.S. And in fact, that CDC testing has shown that people's blood levels are going down for these long-chain PFAS chemicals. However, they do tend to stick around in our bodies for years. They have what we call a human half-life of years in our bodies. So that means even after you stop being exposed to any more PFAS or PFOA, it will still take years for the amount in your body to go down by half. Now, PFAS will still continue to be in our environment for a long time to come, and current use products contain different PFAS chemicals. So instead of stopping the use of PFAS altogether, manufacturers are now switching to other PFAS. Sometimes these are shorter chain cousins of the long chain PFAS that they phased out, and there are other variations of the chemistry as well. We don't know a whole lot about these replacement chemicals, but for some of the newer replacements, we know that they can stay in our bodies for weeks to months, which is somewhat of an improvement over the long-chain PFAS, but still is an extended amount of time. Exposures to PFAS, PFOA, and some other PFAS chemicals have been linked to a range of harmful health effects, including effects on the immune system, increased cholesterol and other effects on metabolism, thyroid disease, decreased birth weight, testicular and kidney cancer, and in laboratory studies, altered mammary gland development. And this is of concern to us at Silent Spring Institute because we want to know more about environmental causes of breast cancer. And we think that chemicals that can alter mammary gland development might increase breast cancer risk later in life. For today, I'm going to focus more specifically on immunotoxicity. And again, I'll preface this by saying I'm not an immunotoxicologist. If you end up having some more detailed questions about exactly how the immune system works, I'd be happy to write those down and connect you to other experts who study these kinds of questions in more depth. First, though, I wanted to step back and talk about how do we learn about toxic chemicals? How do we know which ones are harmful to our health and what the health effects are? One way that we can learn about toxic chemicals is through epidemiological studies, or sometimes we just say epi studies because that's easier to say. So these are studies in people. These can be studies in communities that have had high levels of exposure. And if you've seen Dark Waters or the documentary film, The Devil We Know, you'll know that that focused on a community in West Virginia and Ohio where the levels of exposure to PFOA were highly elevated due to the proximity to a DuPont plant. Other epidemiological studies can be conducted in the general population. for instance, for PFAS, because for PFAS, because so many people are exposed, some studies do look at effects in the general population. So the benefits of epi studies, obviously they're directly relevant to people because they're based in people. So they account for human physiology. However, there are some limitations to epi studies as well. It can be difficult to link health effects to one specific chemical. People are complicated and we move around and we have different jobs and we are exposed to mixtures of chemicals. So for instance, if we see an association between exposure to PFAS and a certain health outcome, we may have a hard time knowing that it was the PFAS alone that caused that health effect. In addition, epi studies take a very long time, they're very expensive, and they're not well suited for screening chemicals before they go into use because by their nature, they are conducted after people have already been exposed. So they're not very good from a precautionary or preventative point of view. A second type of study is called a toxicological study. So these are studies conducted in laboratory animals, often rats and mice. The benefits of toxicological studies are that they allow us to directly link health effects to one specific chemical or to a mixture of chemicals because we can control all the factors in each animal's life and adjust the dose to a certain chemical. It also allows us to study multiple generations. There's a lot of concern and interest in early life exposures and intergenerational effects. So female animals can be exposed to chemicals, and then the scientists can study how their offspring or later generations may be affected by those chemical exposures. There are some drawbacks. One is that the physiology of rodents may be different than people. For instance, while PFAS and PFOA have a half-life of multiple years in people, they're actually cleared pretty quickly by laboratory animals. So that tells us that there's some differences in how PFAS accumulate and may affect rodent bodies compared to people. And while toxicological studies are quicker than epi studies, they still take a long time and they're still expensive. And then a third type of study I wanted to talk about are called in vitro studies. So these are studies based in the laboratory that use, and they're often based on having certain types of cells in a dish. And so let's say we want to know whether a chemical is, can mimic estrogen in our body. We can have cells that have estrogen receptors and look to see how well a chemical binds to those receptors. So the advantages of in vitro studies are that they allow us to evaluate many chemicals, different concentrations and in different mixtures, and we can get the results really quickly. The obvious limitation is that it's harder to link what happens to one cell or a set of cells in a dish in the lab to the complex interactions that happen within people's bodies. So when we look across all three types of tests, we see that they all have their advantages and disadvantages, but really combining evidence from multiple types of studies can strengthen our confidence in understanding a chemical's toxicity and relevance for human health. So now I'll talk more specifically about immunotoxicity, so how chemicals may affect the immune system. So there's a normal balance of immune function in our body that can be both increased or decreased in harmful ways. So when the response of the immune system is less than what we'd expect, that's called immune suppression. So that means the reduced ability of the immune system to respond to a challenge from a level that's considered normal. This could be mild. It could be a slightly reduced response to a vaccination that doesn't really affect disease resistance. It could also be much more severe and increase susceptibility to common and opportunistic pathogens, as well as to certain cancers. On the flip side, we can also have immune enhancement. So this is inappropriate immune responses to common substances, such as allergic hypersensitivity or responses to self-antigens. So if you're like me, this time of year, I have really bad allergies. And the reason for those allergies is that my immune system is overreacting to some type of pollen that's common this type of year. Other types of conditions related to immune enhancement are related to autoimmunity, where the body is actually turning on the tissues and cells within the body itself. So I'm not going to get into the complexities of how the immune system works, but I just wanted to point out a couple of important features of the immune system. So one is to introduce the term antigens. So these are foreign substances that can trigger antibody production. That actually antigen comes from antibody generator. And some examples include bacteria, viruses, pollen, and dust. I also wanted to introduce antibodies, and these are Y-shaped proteins produced by B cells in the immune system, and these can recognize antigens and neutralize them. So they're very specific to certain molecules on the antigens, and this is an important part of our immune system. And for instance, when we get a vaccination, the goal is to boost our antibody levels so that if we get exposed to a pathogen, our body is ready to fight it off. So measuring antibody production is a sensitive measure of immunosuppression. And we can measure antibody production both in laboratory animals and in epidemiological studies. So in laboratory animals, scientists can conduct a test called the T-cell dependent antibody response. So this is considered the gold standard for evaluating immunosuppression because it's able to look for the association between a certain antigen and antibody production, and that captures all the different steps along the way in the immune system. In epidemiological studies, we can measure antibody responses to vaccinations. It's kind of like a natural experiment. Everyone gets these vaccinations, they get exposed to a known amount of an antigen, and by measuring the antibody response, that's another sensitive way for us to test for immunosuppression in people. Getting back to PFAS specifically, I wanted to spend some time talking about the key findings from a systematic review conducted by the National Toxicology Program in 2016. The NTP reviewed 153 studies, including 33 studies in people, 93 animal studies, and 27 in vitro and mechanistic studies. For their study, they only looked at PFAS and PFOA, and their key finding was that PFOA and PFAS are both presumed to be an immune hazard to humans. So talking about their key findings, the NTP concluded that there was the strongest evidence of immunotoxicity based on the suppressed production of antibodies. This includes both antibody production in laboratory animals and suppressed response to vaccinations. There were some other immune-related outcomes for which there was weaker evidence based on the review conducted by NTP. There was some evidence for increased rates of hypersensitivity-related outcomes. So that's more on the immune enhancement side. In people, this can include conditions like asthma, allergic responses, and rhinitis, which is runny nose, sneezing, and stuffiness for PFOA. Some other health outcomes include increased occurrence of infectious diseases such as cold, flu, gastroenteritis, and ear infections, increased rates of autoimmune diseases like ulcerative colitis, and suppressed activity of natural killer cells. So these are immune system cells that are important for warding off viruses and cancer cells. Just to give you a sense of the outcomes of some of the EPI studies that were included in the NTP Systematic Review, I'll talk through a few examples. One is one of a series of studies conducted by our friend and colleague Philippe Grandjean from the Harvard School of Public Health. He's been studying children in the Faroe Islands for many years, and his work was some of the earliest to show impaired vaccine response in children. So for instance, in 2012, he published a study showing that PFAS and PFOA exposures at age 5 were associated with lower levels of diphtheria and tetanus antibodies in 7-year-olds. Again, this is in the Faroe Islands. This is a community where people can be exposed to elevated levels of PFAS from their diet. Another study was conducted in adults. This was part of the broader C8 study that I mentioned in the community near the DuPont plant in West Virginia. This epidemiological study included 69,000 people who'd been exposed to contaminated water from the DuPont plant. And in this study, they focused specifically on PFOA, which is sometimes called C8, and they found higher levels of PFOA were linked to lower levels of influenza antibodies and an increased risk of not attaining a threshold level that's considered adequate to offer long-term protection. So those were two studies in communities that had elevated exposures. Other studies have looked in the general population. So the study in Norway in 2013 looked at 3-year-olds and found that higher PFAS exposures were linked to lower levels of rubella antibodies after vaccination, more frequent colds, and more frequent episodes of gastroenteritis. Another study in Denmark, also looking at 3-year-olds, found associations between PFAS exposures and more frequent fevers, as well as more frequent fevers with cough and nasal discharge, with fevers being a marker of infection, a sensitive marker of infection. And finally, I wanted to mention a study here in the US of 12 to 19-year-olds. This was based on an analysis of data collected by the CDC as part of their national testing program called NHANES. And Stein et al. Concluded that higher levels of PFAS were linked to lower levels of mumps and rubella antibodies. And I just wanted to highlight that this study and the prior two are noteworthy because these are not conducted in areas that have highly elevated exposures, but were found in people with PFAS blood levels consistent with the general population. There have been numerous studies in laboratory animals as well. I won't go into great depth on these, but I just wanted to highlight two that were some of the earliest works showing conclusively that similar to the decreased vaccine response in people, that PFAS and PFOA can also suppress that antibody response, that T-cell dependent antibody response in mice. And this PFAS study conducted by Dong et al. In 2009 was used by New Jersey as the basis for their PFAS drinking water standard. All right, so just to wrap up on immune system effects, so immune suppression, as measured by decreased antibody response, has been linked to PFAS exposures, both in laboratory studies in animals as well as in people. Some of these studies have been conducted in more highly exposed communities, while others have been based on the general population. There's been some evidence, but less consistent evidence for other types of health outcomes, such as asthma or ulcerative colitis. I also wanted to note that most studies have focused just on a handful of PFAS chemicals, yet as I mentioned at the beginning, there are thousands of different PFAS chemicals, so there are concerns that there might be exposures to other PFAS that we are not yet studying that might also be affecting immune health. So I wanted to end by talking about why scientists are concerned about potential links between PFAS exposure and COVID. So the ATSDR, this is part of the Centers for Disease Control and Prevention. ATSDR is the Agency for Toxic Substances and Disease Registry. And back over the summer, they came out with a statement on the potential intersection between PFAS exposure and COVID-19. They acknowledged concerns from people, especially those in communities that have had highly elevated exposures. They acknowledged concerns that the PFAS exposure might affect their risk of COVID-19. They acknowledged the strong body of evidence showing links between PFAS exposures and effects on the immune system, and they noted that more research is needed to understand these potential links. This is also a topic that has certainly gotten a lot of attention and gotten a lot of people wondering, again, especially in communities that have had a history of high levels of PFAS exposure. So there are some key questions as we consider how PFAS might be related to COVID. So one key question is whether PFAS exposures could reduce people's resistance to COVID, just like we saw decreased resistance to other infectious diseases, such that people who do get exposed are more likely to become ill or to develop severe symptoms. On the flip side, when we think about the potential for immune enhancement, scientists wonder whether PFAS could contribute to overreaction to the virus by the immune system. So for instance, the main concern and the reason for such severe symptoms in people who have COVID is called a cytokine storm. So this is another part of the immune system associated with autoimmune diseases where the body can attack its own cells and tissues and not just fight off the virus. So there's questions whether PFAS could exacerbate that response. Finally, we all have our fingers crossed that there will be a COVID vaccine coming soon. There are concerns, again, especially among people who've had high exposures, about whether their body's ability to produce antibodies in response to the COVID vaccine might be reduced. So what is being done to address this? Well, the NIH has a number of funding opportunities, so scientists are ramping up efforts to address these questions. The CDC is finding ways to incorporate PFAS into some of their COVID studies, and researchers who are conducting studies in PFAS-affected communities are considering how best to incorporate COVID. And I also wanted to point out special considerations for vulnerable communities that may have high PFAS exposures and other risk factors that could multiply and exacerbate the effects and the vulnerability of these communities to COVID. So if you'd like to learn more about our research or about PFAS in general, I'll point you to a couple of resources. So one is the PFAS Exchange. This was developed by our PFAS Reach study, which is a partnership led by Silent Spring Institute, along with Northeastern University, Michigan State, Massachusetts Breast Cancer Coalition, Toxic Section Center, now called Community Action Works, and Testing for Peas. We have a new fact sheet that we've just put online about PFAS in the immune system. We have other types of fact sheets, resources for the general public, and especially for PFAS-affected communities, and a data interpretation tool to help people make sense of PFAS water and blood test results. Silent Spring Institute is also a research partner on the STEEP Superfund Research Program. This is led by University of Rhode Island and includes Harvard and Silent Spring. We're partnering with Mass Breast Cancer Coalition and the Sierra Club for our community engaged research on Cape Cod. And the STEEP website has great resources and videos to talk about our research, PFAS health effects, and to give tips to reduce exposures to PFAS. So with that, I'd like to thank you again for tuning in. Thanks to Mass Breast Cancer Coalition for inviting me and for all the important work that you do to raise awareness about chemical exposures and links to breast cancer and other disease. And here are some resources if you'd like to learn more. Thank you.
Wonderful. Thank you so much. We do have several questions that have come in. First, current PFAS exposures are measured in the blood, will not provide information on historical exposures. So how is that addressed?
So that's a great question. And you're right that a current blood measurement will provide some indication of exposures in recent years, but not a cumulative lifetime exposure. This is something that we're considering right now as part of our PFAS health study. It's called the Massachusetts PFAS in Your Health Study. And this is a part of a multi-site study that's being led by the CDC and ATSDR in communities in 7 states across the country. And for this study, we are assessing PFAS exposures in 2 ways. One is by measuring PFAS in blood, but another is by reconstructing past exposures. So asking about where people lived over time, asking about their water consumption patterns, and we will also be learning more to try to estimate what PFAS levels were historically in the public water supplies. We're also doing this as part of our PFAS Reach study as well, so that we'll have 2 independent measures of PFAS over time in people's lives.
And can you speak to, can PFAS chemicals in the body increase the likelihood of delayed COVID responses?
That's a great question. I don't know that anyone has answered that question yet. I'd be happy to try to follow up with other experts, but I'm not aware of any studies that have answered that question yet.
Okay. And how is the science around PFAS and the studies that are being done, how is that trickling down to your day-to-day general practitioners, so that when people are seeing their doctors, is this information the kinds of things that are starting to filter through to general practitioners so that they're speaking to their patients about exposure and prevention measures?
I would say for the most part, no. I think this is a huge gap. Environmental health is not really covered that well in traditional medical education. It is something that we aim to address through our PFAS exchange. We're working with doctors to come up with resources that will be helpful for medical professionals to give guidance to people who've had high exposures. But it's definitely a need that we hear, particularly in PFAS-affected communities. When people learn about PFAS in their water, maybe they have blood test results that show elevated exposures. Doctors often aren't familiar with PFAS and don't know much about health effects. And one need that I hear articulated in many communities is the need for medical monitoring. So you may know that you've had high exposures. You've heard that there are many health outcomes that might be linked to that exposure. And people often want enhanced monitoring for certain health outcomes that might, be more closely related to their elevated PFAS exposures, and have that be part of their medical history, so that if there are any symptoms or conditions down the road, to help their doctors maybe zero in on, diseases that might be related to PFAS exposure. But I do think it's an important need because a lot of doctors just aren't that familiar with PFAS. So we want to create more resources to help inform them.
I've got a couple of questions around the same topic, so I'm going to try and combine this. Do you see that the exposures are coming more through people's consumption of certain foods or through the drinking water? And is that even a question that can be quantified?
It depends where you live. So in an area that has elevated PFAS in the drinking water, it's likely that will be the largest source of exposure. Now you might ask how elevated is elevated. And what comes to my mind are guidance documents developed by the state of New Jersey as they develop their drinking water standards. And they showed that somewhere between about 10 and 70 parts per trillion, that drinking water becomes the dominant source of exposure. So, and this can certainly vary over time as well. So, for instance, I think of Hyannis, where two of our health studies are based. Once the high levels of PFAS in the water were discovered, the water system installed treatment and is purchasing water from other systems, and the PFAS levels are now drastically lower than they used to be. So for people living in that community and in others, in the past, their major sources of exposure may have been from the water, but moving forward, other sources of exposure, more typical of the general population, are likely becoming more important. So in the general population, there's actually not a lot of great information to know the relative contributions of food versus consumer products, and within food to know how much comes from the food itself versus packaging. There have been studies showing links between fish consumption and PFAS levels. Some of our work showed that microwave popcorn consumption was associated with higher PFAS levels, and that people who ate at home more seem to have lower levels, suggesting a role of packaging. So we know that there can be significant contributions from food, but it's difficult to put all the pieces together, because we know if you have, say, a new Scotch-guarded carpet, maybe that might be a larger source of exposure than from food. So I guess the short answer is it depends a lot on where you live, and recognizing that those relative contributions can change over time.
Great, thank you. Is there evidence that PFAS interact with each other to influence immune outcomes?
So the issue of mixtures is really a huge unknown. And I don't know the answer to that off the top of my head, to be honest. I know that certain agencies, such as the European Food Safety Authority, are now lumping together multiple different PFAS and assuming that they are similarly toxic. So they're combining the total amount of 4 PFAS chemicals and developing recommended limits for PFAS in food. Here in Massachusetts, the guideline or the new standard is based on the total amount of 6. Some of those we have a lot of direct evidence for, and some Mass DEP made some precautionary assumptions based on the similarity in chemical structures. But I think that's still really an area for research to know whether multiple PFAS effects add up in a linear way, whether they can amplify each other's effects, or whether they compete in some more complicated way.
And when PFAS is removed from drinking water, what happens to the material that was used for the removal process? How is that disposed of?
That's a great question. So that's, these are forever chemicals. So you can remove them from drinking water, and then what? So the most common way to remove PFAS from drinking water is with activated carbon. So basically like a 20-ton Brita filter to remove PFAS from the drinking water. There are other techniques as well, but activated carbon is a common one, and it creates a huge waste problem. So PFAS are extremely persistent under normal conditions. They do break down at very high temperatures, and so some waste and some of the activated carbon media get incinerated. The incineration of the carbon can regenerate the surfaces and allow it to be reused for more water treatment. I know here in Massachusetts, the state has had a program to collect firefighting foam that has the long chain PFAS, and I believe that is also being incinerated. And it sounds good. If you have high temperatures, you can fully break down the PFAS. That seems like it could be a good thing. But there are huge concerns, especially in communities around these incinerators, because if the temperature isn't quite hot enough, there are concerns that the PFAS won't actually break down, or might just break down into smaller PFAS, or that there might be other byproducts. And it also raises concerns about sort of moving that much PFAS-containing material around. So there's really no easy solution, but incineration is, I guess, the most common answer, but not an easy one.
And you had mentioned that obviously there are thousands of PFAS, and only a few are being regulated at this time. How were those selected over others? How was it determined those were the ones to zero in on first? And what's the likelihood of expanding beyond that initial base?
Mm-hmm. So PFAS and PFOA have gotten the most attention because, well, they've tended to show up most frequently in water and in people's blood samples. And we have the strongest evidence of harmful health effects. They were discovered earlier, and they show up more frequently and have been studied more. And there are a handful of other, mostly long chain PFAS that have also been studied. The ATSDR released a draft toxicological profile, and they came up with quantitative risk levels for PFAS, PFOA, and 2 other PFAS chemicals. But what about all the other PFAS? That's a great question. Some of them are newer chemicals because they're more replacement chemicals, so they haven't been studied as long. What we know from early evidence of toxicity testing is that the newer chemicals seem to show some of the same health outcomes, but they might occur at higher levels of exposure. So the long chain compounds like PFAS and PFOA seem to cause effects at the lowest levels. I would note, though, that this can change over time. The FDA recently took back approval for a short chain PFAS chemical that they used to approve for the use in food contact materials, and based on evidence from their own scientists finding this chemical was more toxic and more bioaccumulative than they predicted, they took back that approval. So that, our list of chemicals of concern is kind of evolving. There are some challenges in terms of measuring more PFAS chemicals. So right now, the EPA has a method that includes between 20 and 30 PFAS chemicals. So there are a lot that just aren't being measured. And then there are chemicals that don't seem to accumulate in blood. So even in a community in North Carolina, where Gen X, which is a replacement for PFOA, where that was a chemical that was at high levels in the environment, people didn't seem to accumulate high levels of that chemical in their blood. They accumulated other ones. And there's some thought that different PFAS may behave differently in our bodies. So blood serum, which is what people typically measure, might not be the best way to evaluate exposures for other PFAS. So looking at whole blood or looking at urine might be a better way to look at exposures to these other PFAS chemicals. And there's increasing interest in methods that can look at total organofluorine. So rather than trying to figure out the identity for each individual chemical, and the chemists don't even know all the chemicals that might be there. And if we look at the total organofluorine, that might give us some sense of the total amount of PFAS and how much we're missing when we just look at our typical list of 10 or 20 compounds.
And I know you speak of looking for traces in blood and urine. What about fatty tissue?
That's a great question. I could get back to you about that. I don't know a lot about that, but I do know that PFAS, so other types of chemicals like PCBs tend to accumulate in fat. And my understanding is PFAS actually tend more to bind to proteins. So they accumulate in blood because they can bind to proteins in our blood. So I'm not sure about whether measuring fatty tissues would tell us something different, but that's a great question. I don't totally know the answer to that.
Checking to see if we had any more questions. Well, Dr. Schaider, I want to thank you very much on behalf of Cheryl Osimo, MBCC's Executive Director, and our Board of Directors. I really want to thank you for sharing your insights on PFAS with our listeners today. And I want to thank all of our listeners for joining us and letting you all know that anyone who is interested in a recording of this webinar, that it will be available later today on the MBCC website. And as Dr. Schaider referenced, the archives from past PFAS webinars are also on our website, and I invite you to visit and learn more at mbcc.org. Thank you all very much. Dr. Schaider, thank you again, and I wish you all a good afternoon.
Thank you.